Here with Professor Peter Rich from the Glynn Laboratory of Bioenergetics at UCL. Professor Rich, on March 9th, 2016, you gave a lecture at New York University in London on the topic of your predecessor, I believe, Professor or Dr. Peter Mitchell, and his revolutionary chemiosmotic theory. This was to commemorate the 50th anniversary of the publication of Dr. Mitchell's independently published papers, his gray books. So you first joined the lab when it was still an independent institution at the Glynn House in Cornwall. What brought you to the Glynn Lab in the first place?
At that time I was in Cambridge working on quinones, and I was heavily involved in trying to understand the relation between electrochemistry and biochemistry and chemistry of quinones. And the area was a bit messy at the time. And there was a meeting there, and Peter Mitchell came up. I had a poster actually on this stuff that nobody was interested in at all, because it was a bit offline. Except Peter Mitchell, who came up and saw this stuff, and he realised that it related to some of the stuff that he was trying to figure out with quinones. So he invited me to go down there, which I did a few times. And then he invited me to go down and take over the research labs there. So that was a big decision, but decided to be kind of fun. so I left Cambridge and took over running the labs there.
And then in 1997, you returned to UCL.
Yeah.
And so you chose to keep the name Glynn for the research group when you officially returned to academia. Why was that?
Just to keep the continuity, really, of the labs, because we were still working on the same types of problems, and I was then well known for being associated with the Glynn Laboratory, so I decided to call the unit here the Glynn Laboratory within UCL, just to keep historical continuity, really, more than anything.
And in your lecture, you mentioned how Dr. Mitchell was ostracized by the scientific community, and he decided to establish himself in Glynn House. But do you think that the location played a role in the science that was produced there?
Yeah, that's a good question. Actually, Peter Mitchell had many interests, one of which was, in addition to science, was doing up old houses, and he'd already done up some old houses, and he certainly liked to be very independent and outside of the university system. So he was offered the house in Cornwall very cheaply. It had run down actually after the Second World War. It sort of got dilapidated, so he was offered it very cheaply. Since he liked doing up places and he was looking for a quiet place to live outside the university, he bought that place and did it up. And yeah, I think it probably did help in his case, yes, because he did need quiet. He always said that universities were a very low signal to noise place for doing research. There's lots of students and bureaucracy and all, endless meetings.
So with that, you have, at Glynn House you would have the independence and the quiet. Did you not feel the same way? And is that why you just decided to come back to London?
No, I liked it. I thought it was a very efficient way to do science, actually. Of course, it did have negative points to it as well, because we were a long way from anywhere.
Right.
So we wouldn't have other experts on our doorstep, though people would have to have a reason to come to the place.
True.
But in terms of doing research, yes, we had a very small but very focused and expert and very stable group of scientists there. So it was a very effective way to do science. And I would have continued it there if it was economically viable to do so. But it was more economics rather than science, which is why I eventually moved the place.
Was it also economics that you chose to return to UCL instead of back to Cambridge?
That was a difficult decision, actually, yeah, as I had offers from both to go back to Cambridge and to come to UCL, and there was a variety of reasons for not going back to Cambridge. In retrospect, I probably should have gone back there, because as I kind of fitted in, because I knew everybody there, and fitted in well. But also I wanted to be able to travel back to Cornwall regularly, and going from Cambridge to Cornwall,
Right.
you could hardly do it in a weekend because of the extra travel needed, whereas from London it was much easier. And they offered me a good position and good resources here at UCL, and I did know quite a few people here, so I decided to move it into here.
Is it a choice you regret, or since you said you should have gone to Cambridge?
It would have been nice to have been back in Cambridge, apart from the fact that it's too far away from Cornwall.
Yeah.
And the academic environment in Cambridge I like a lot. So yeah, in retrospect, I was always on wavering on that decision actually, but no, I can't say I regret it. You know, one door closes, another one opens. We still managed to do effective research.
I have a few questions on Peter Mitchell, because as you mentioned, he was like the houses, and he ran his own dairy farm successfully and minted silver. So he seemed like a well-rounded person. Do you think that influenced the science he did, or is that a trait that you would say is very important in a scientist?
Yeah, I was asked this recently while I was in Japan having to give a talk. What makes a successful scientist? I'm not really sure what the answer is to that, but of the very successful scientists that I've known, they've always brought something else to the table in addition to the science itself. And in Peter Mitchell's case, that extra bit was probably, he had a very abstract philosophical view of the world and of science, and the way that he did science was in a sort of abstract formulation sort of way, which was partly responsible why nobody could understand what he was talking about at the time from the way he presented things. So yes, definitely. I think for all successful scientists, it's part of your life. So your character permeates the way that you do the science. And to do something a bit different, you do need a different angle, which can be any of a number of different things. I think I gave the example of another famous scientist that I knew extremely well, Robin Hill, and he was an expert in vegetable dyeing, and he used to make all his own dyes from vegetables that he grew, plants that he grew, because he was a watercolour expert, an expert painter, and in fact his paintings are quite valuable. But his knowledge of dyes all goes into redox chemistry. Dyes are redox chemicals. Any dye can go oxidized and reduced, and the redox process is part of the thing. And that really helped him to discover his famous Hill reaction, which is one of the basic cornerstones of understanding photosynthesis. So that's another example where he brought something different to the table.
In these kind of skills, were they more part of the person, so Peter Mitchell, or was it actually integrated in the whole life there in the lab work?
I think for most very successful scientists I know, science and life generally are completely integrated. That doesn't mean that people are obsessively doing science all the time, but people's characters go into the way that they do the science.
Who are some of Peter Mitchell's most important influences in terms of leading to the theory and generally?
Oh yeah, that's a very controversial point. Whole books have been written about that. What would Peter Mitchell say? One of the great influences was somebody called David Kaelin, who was before my time in Cambridge, and actually Peter Mitchell was considered to be very arrogant as a student, and he didn't get on too well in the biochemistry department, and David Kaelin actually took him on, and that's when he got set up with his lifelong experimental collaborator Jennifer Moyle. They were put together in a room, not in biochemistry, in the Parasitology Institute next door. So he was definitely a major influence, and also Kaelin was the person, in my opinion, who actually discovered the respiratory chain effectively. very dangerous thing saying who did what when in science. So there was the respiratory chain development going on, and membranes were being properly discovered and understood at the time. And in other places, people were beginning to understand the nature of mitochondria. When Kaelin discovered the respiratory chain, mitochondria weren't properly known. But I would say, being in Kaelin's environment there, and so there was someone there called Danieli who was discovering the lipid bilayer membrane, and they were working on bacterial transport processes, other people there. So I would say the influences around him in Cambridge were probably the most important.
In also probably some of the works you've mentioned, it's been pointed out that Peter Mitchell was very firm in his opinion about his own research, and didn't always like other researchers suggesting amendments and other mechanisms. One example is the cytochrome oxidase as a proton pump, as suggested by Morten Wikström.
Oh yeah.
So was it sometimes difficult working and discussing findings from other labs with him, like as a personal experience for you?
From a personal experience, I didn't find that at all.
Okay.
He was extremely open to things. And if you take that particular example with Morten Wikström, I've been for the last week with Morten Wikström actually, 'cause I've been running a meeting in Austria. I only came back yesterday. Peter Mitchell eventually admitted that he was responsible for holding up the field of cytochrome oxidase more than anybody else by not accepting that. And the reason he didn't accept it was, first of all, the experiments are very difficult to do to show this extra coupling process, which Morten Wikström had actually shown. And so Peter Mitchell didn't really do any hands-on experiments very much. They were all done by Jennifer Moyle. And the reason that Peter couldn't accept the idea that it was moving these extra protons is because he couldn't think of an abstract mechanism based upon the chemiosmotic principles by which the thing occurred. And actually, when I went to Glyn, it was just at the time when he just accepted that there was this extra function, and the reason he accepted it actually is because he'd come up with an abstract chemiosmotic mechanism by which it might occur. Although actually the first thing I did when I got there was to prove that that wasn't the case. It couldn't work by the mechanism that he wanted.
So
But he was very open to that.
how come then that you say he was very open, but then a lot of people would argue the opposite, like Wikström said that it sometimes was kind of challenging to, where he wouldn't look at the experimental evidence and say like, no, this is impossible, maybe because he didn't have an abstract mechanism, but
Yeah.
how come there were so many different views on
There
him?
were a lot of strong characters in the field at the time, so he'd already had a massive battle by that point in order to get recognised with his stuff, and as I said, he'd got kind of rejected almost from the scientific community at the early stages, and so he did have to fight incredibly hard and maintain a position of argument, of which there were many people arguing against it or even dismissing it completely. So I think that the case with Morten Wikström is a slightly more special case, actually. They did do a lot of careful work, and it's true that initially Peter Mitchell explained the Wikström phenomenon as being a complex artifact of very complex experiments, and Jennifer Moyle then worked on this really for several years and slowly came round to the idea that in fact it wasn't an artifact. And as I say, it was. I think there was bad feeling there at the time. That was actually slightly before my time, because I was involved in this quinone stuff in Cambridge at that time. I wasn't involved in that controversy. By the time I got to Glyn, he'd already come to an agreement that Wikström's theory was right, and was very generous about it then, actually. And I think they got on reasonably well after that.
So you would still say that there was sufficient freedom for his like students or Moyle or like everybody working in his lab, or was he very
Well, you
directing?
say everybody. There were very few people working
True.
there. I mean, he was very different to the normal scientists, and he worked with a very small number of people. When I arrived there, there was Peter Mitchell. Jennifer Moyle actually had retired by then. There was one postdoc, no students, no PhD students, 3 technicians, 3 long-term technicians, and 1 postdoc, and Peter Mitchell, and a secretary.
So he was
so-
mostly deciding what was happening.
Well, when I got there, I had complete freedom to do anything at all. I mean, we often discuss things, but there was never any question about even trying to direct me in any direction. And as I say, the first thing I did was to disprove his theory of how cytochrome oxidase worked, and that was fine. That worked fine. And from there, actually, we went on to do some experiments eventually, where we were looking for something which we both thought was a good idea, which turned out to be wrong. But because it was wrong, it gave us an insight into how the mechanism actually works. It's quite good when you get something wrong, because it means it's showing you that you're not thinking about it the right way. When you get an anomaly, as long as it's not an artifact, anomalies are really good. If you just do an experiment and it shows you what you thought it was going to do, it's not very exciting in a way.
And so, because of this research and the establishment of the theory, Peter Mitchell was awarded the Nobel Prize in Chemistry in 1978, and he was the sole recipient, which is actually rare,
Yes.
and kind of demonstrates how important and revolutionary the theory was for the field of modern biochemistry, but also, I guess, physics and biology and chemistry. But there was another person in the lab, as you mentioned several times. Jennifer Moyle did a lot of research, and she was a great scientist. So how come she didn't share that prize?
Yeah, well, that's a good question too. But she was she wasn't a theoretician in any way whatsoever, and she would have agreed as well that she didn't contribute to the formulation of the chemiosmotic theory. She was just a fantastic experimentalist, but was not a theoretical thinker at all. So I think it was probably fair that she didn't share the Nobel Prize. Of course, some other people thought they should have shared the Nobel Prize with him, as with all important bits of science, history is complicated and often gets confused.
But considering how long she was there, do you think the theory would have been possible without her?
The theory would definitely have happened without her,
Okay.
but the experimental confirmation that came from their own work wouldn't have come, because Peter Mitchell was not really an experimental person.
So
He was very good at looking at other people's data and sort of interpreting it and seeing its strengths and weaknesses, but he wasn't really an experimentalist at all.
but Moyle is kind of still very unknown today. So do you think that's basically because of that she was more of an experimentalist, or are there other factors contributing?
She didn't usually go to the conferences at all.
Okay.
So she was never there arguing the case, and I think that was her own choice. I'd say she'd retired from Glyn and moved back to her home city of Norwich by the time I'd gotten there. Although she was still visited the lab quite often, but I think it was her choice to be the experimentalist and not to try to push the theory. So, for example, Peter Mitchell's grey books, the grey books that they're called, the ones which he published privately, are just his own authorship of stuff, and Jennifer didn't really contribute to those.
And with just quick question about also the guy, hasn't everything he set up the fact that he was able to become independent from Cambridge and Edinburgh, do you think that's also related to that he had a large inheritance? Do you think it would have been possible without the means around it?
It would have been impossible without the inheritance,
Okay.
for sure. So it's very expensive to set up a lab, particularly if you're not getting funding from the normal funding agencies, which he didn't get. So, that it would never have happened.
Did he support other projects afterwards in terms of that seemed promising, which had similar characteristics as how he started out? Or
There was never enough money for that.
Okay, yeah.
The place was always on the edge financially. Because he did have an inheritance, and he bought the place and did the place up. But for at least the first 10 years, he was unable to get a research grant from anywhere because he was so outside the scientific community. So that really used up all of the capital. And the Mitchell family would sort of chip in some core money every now and again to keep the place going. It was always kind of on the edge. So there wasn't money to support. That was the idea originally, to set up a research foundation which could support research generally, which the standard academic system might not want to support. But it never got to the stage of having enough money to give money to someone else. It always needed money.
And finally, what do you believe is the most important legacy of Peter Mitchell today? Like, what should we take away from him as a younger generation, for example?
Well, of course, the legacy is his chemiosmotic theory and the way of thinking about things. If you want to do something which is at the edge between chemistry and physics and biology, right, which is what that was, you have to be completely comfortable with all of the basics. So you have to fully understand all of the basics, not just as a student, just being able to write down the equations, but understanding where they come from and what they mean. And once you're comfortable with all those things, you can then manipulate them as you want to produce some new kind of synthesis. So I think he was a fantastic example of that. I've got lots of his textbooks here. And you'll see they're all on things like there's an R.P. Bell, the proton, Guggenheim thermodynamics, physical chemistry of liquids and solids, the theory of diffusion, Planck's theory of radiation, all those sorts of things. And once you're comfortable with all those things, you're fantastically well equipped to do something of significance. And it's not that it's that difficult. It's just that you've got to have a desire to do it. And complicated equations. I keep saying to students, complicated equations are actually just lots of simple equations all stuck together. And the bit you'd really need to understand is not the complicated equation, but the simple one.
Yeah.
Right? Newton's laws of motion. Everyone can write down Newton's equation, but what it actually means is that you've got to sort of get a grip on that, and that applies to all these things. So complicated equations, as I say, really it's true. They're all just simple things, really, but a lot of them in some cases stuck onto each other. So for me, that was a very guiding principle, to be able to make sure you understand fully what it is you're talking about.
Do you think that current institutions, like for example UCL, are doing a good job on this? In terms of
I think it's much harder. I think it is much harder now. I mean, science is so complicated. You take the biological sciences. Each area of biological science has got so much information now. I sympathise with students. It'd be very hard to get your head around that stuff. When I first went to Glyn, because I was already sort of an established scientist by then, but I was working on quinone chemistry, and I decided to get involved in this cytochrome oxidase story. It took me 2 years before I felt fully comfortable with the literature, that I knew what was correct and what was not correct in that. And I had a lot of time to read, because That was 2 years with a lot of reading time because I was then in Glyn, so there wasn't any pressure to do it. So 1 day per week I was devoted to just reading and thinking. That's almost impossible in a university nowadays for people. So I think it is much harder. I think UCL's doing a pretty good job on its teaching, but there's too much stuff out there.
So less stuff and more reading time.
It depends what you're trying to do, though.
Yeah.
For example, if you want to do just if you want to be and I don't want to be rude about molecular biologists, there are 2 types of molecular biology. By molecular biology, I mean messing around with genes. You can be someone who does the molecular biology transformations, and that really is just cookery. Okay, cookery with attention to detail, but there's no intellectual effort really required in it. On the other hand, if you want to invent new methods of doing molecular biology, like Fred Sanger's, okay, that is a different kettle of fish. That's where you need to go back to the basics, where you kind of understand everything. You don't need to understand anything to do molecular biology. You could go in there and having done none of it, as long as you follow the recipe precisely, you will get the result without even knowing what you're doing. And that's a very valuable thing in itself. But that's a different type of science. So I think it depends, what you want, what you're aiming to do. But if you want to discover something truly new, it's probably not going to work just following recipes.
Right, so we as the younger generation were introduced to the basics, at least, of chemiosmotic theory as part of our secondary school education. And because of this, it's partially hard to understand why it was so controversial in its early days, since it's not something we really question now.
Yeah, that's true.
So what do you think is the most groundbreaking or counterintuitive aspect of this theory?
Well, the main thing at the time was that all biochemical transformations were considered to occur by, particularly say for ATP synthesis or something, by so-called group transfer on protein. So the way you made anything was you got the various functional groups to be brought together on the same enzyme. So in terms of ATP synthesis, and it was already shown that some of the enzymes, for example, of glycolysis, which do produce ATP, for example, phosphoenol pyruvate, the pyruvate reaction, that is a classical group transfer thing where you've got a phosphate group stuck on the protein and an ADP stuck here, and the protein undergoes a conformational change and you get the phosphate bond formed, and then it leaves, just like all other enzymology at the time. And it was thought that oxidative phosphorylation must surely work by the same way, because there wasn't any other type of mechanism out there at all. And so there were a huge number of people trying to find this active species which had the phosphate in an active form, either on the same enzyme as the ADP, but it had of course to be connected to the electron transfer bit as well. So they're looking for what was called the high energy phosphate intermediate. And this occupied vast numbers of labs with vast amounts of research resources, and of course they never found it. But what Peter Mitchell was saying was that it was anything to do with classical biochemistry, that it was a so-called proton motive force involving, well actually even Peter didn't properly get it right first of all. He thought it was a pH gradient, that's the osmotic bit. Whereas actually there's the electrical gradient as well, which he added later to the thing. But this was completely outside the realms of normal biochemistry, and at that time when he proposed it, even membranes were not properly understood. So in order to have a proton motive force, you have to have a protonic barrier as well. And so it was beyond it was beyond normal biochemistry, and a lot of people had a lot of time and money invested in looking for classical biochemical solution to the thing. So that's really why it was so hard. So there were political reasons and reasons of the nature of the thing. As I say, it was right on the interface between physics and chemistry and biology, and not everybody, in fact very few biochemists, were equipped to understand those three aspects.
And was there any particular inspiration that caused Peter Mitchell to start thinking about ATP synthesis in terms of the proton motive force?
Yeah, that's quite a complicated question. There were several because nothing comes out of nowhere in science. I don't think it does anyway. There's always something preceding it. So it was known that plant roots. So this here, Sir Rutherford Robertson in Australia had worked with several other people, showing that plant roots, when they take up stuff, they move protons out, and these are somehow related to respiration. That you get a pH outside when you start to respire. He didn't figure out in terms of anything to do with energy conservation. But there's also the gastric, the cells of your stomach are somehow pumping protons into your stomach to make your stomach acidic for nutrition, and that has to be driven by electron transfer or by ATP. So there were indications that, and Peter Mitchell had started working on bacterial transport, of how bacteria get stuff into them, and membranes were beginning to be discovered. So there were those indications out there which he was able to bring together, these developing ideas of a membrane, and the fact that definitely some biological systems, when you've got respiration, you've got protons moved. Of course, nobody had any idea about the way that he put it together, but those things were sort of out there. And there was this guy, R.J.P. Williams, Bob Williams, which forms a very complicated and still quite depressing part of the whole story, because Bob Williams, a chemist in Oxford, he had also shown chemically you can make ATP by having a very high proton concentration. So he had already suggested that respiration might produce locally a very high proton concentration somewhere in the membrane, which is not how it works, but that could actually make ATP. So Bob Williams went on to claim that actually he'd invented the chemiosmotic idea from that, and that is open to interpretation and is still argued about even now, but you have to read all the various books about that. So getting back to your question on the Nobel Prize, Bob Williams was certainly one who thought he should have shared the Nobel Prize.
It seemed like a very complex relationship with the letters back and forth.
That's right, yeah. So Bob Williams I knew quite well, actually, and we got on really well until we started talking about anything chemiosmotic. So in my opinion, Bob Williams did fantastic work on the interpretation of NMR of complex proteins, and his work on inorganic biochemistry, roles of metals in biology, which was fantastic work, which he should have got a Nobel Prize for that stuff. But when you look at his stuff related to the chemiosmotic theory, if you read the stuff, and I did look at it closely, there's many good ideas in there, but there's too many ideas. In other words, he's covered almost anything, and you could construe almost anything from the way the things are written. Whereas what Peter Mitchell did was focused on the chemiosmotic idea and then went on to test it and ultimately show that it was correct. Whereas Bob Williams had suggested everything, which was very clever to think of all those things, but he didn't go on to tell us which of the 100 possibilities was the correct one, sort of thing. He wouldn't agree with that, and unfortunately he's not around anymore to argue his case, but that's my opinion of it anyway.
And it has been said that chemiosmotic theory is to biology what Einstein's theory of relativity is to physics, as it revolutionized modern biology. Do you feel that this is an adequate description, and would you say that it is still true today?
It's certainly one of the cornerstone developments for metabolism and energy production. I mean, it is the central idea. Of course, biochemistry, as I said, is a very, very broad subject. So chemiosmotic thinking doesn't have to come into lots of things. So I think that's perhaps going a bit far to do. But yeah, I mean, it was an abstract development which came, which was a unique thing at the time and gave a completely different aspect which had never been thought of properly before. Of course, Peter Mitchell did want to generalise it actually beyond membrane processes to even within enzymes. In fact, we did start sort of looking a little bit at that. Some enzymes have very complicated structures, some have tunnels in them and so on. So the idea is whether you can get chemiosmotic forces within single proteins. I think that still is possible, but that hasn't really been explored properly. So I'm not sure if I'm answering your question there, but yes, it was revolutionary, but maybe not at the same level as Einstein's theory of relativity and so on, because that applies to all of physics, whereas Peter Mitchell's idea applied to a specific but very big area of biochemistry and biology, but not to all of it. Like I say, if you're going to do molecular biology, just gene jockeying, you don't need to know anything about the chemiosmotic theory at all. If you're doing anything with physics, you definitely need to know about Einstein stuff and quantum mechanics.
And it has been said that the chemiosmotic hypothesis was faced with a lot of skepticism from the scientific community at first because it was so counterintuitive. So was there a specific turning point that caused biologists to start becoming less skeptical about these ideas and realize that Dr. Mitchell was onto something that could be potentially groundbreaking?
I think that it happened. It didn't happen in a one sort of thing. It was a sort of slow change of the field. But I think that the photosynthesis field actually was the first to really accept his stuff, rather than the respiratory field, to which he'd written all of most of the stuff anyway. And then the reason being that when you light activate photosynthesis, it was already clear that you actually had you had an electrical process occurring across a membrane, but they didn't understand the proton bit at the time. But when, for example, there was this guy Horst Witt, W-I-T-T, Horst Witt, who had already done experiments where you flash a photosystem and it's quite clear that you get an electric potential across the membrane, although when he did it, membranes were not properly understood. But he realised that there were electrical processes going on. And so the photosynthetic community were much more accepting of Peter Mitchell's ideas. But this comes into politics and sociology as well, you see, because Peter Mitchell didn't work in the photosynthesis area, so he wasn't competing with anybody. Whereas in the mitochondrial area, he was treading on a lot of toes there. And actually the first person to do an experiment to show that you could make ATP with a proton gradient, that was done with chloroplasts. So the first experiment was done in America by someone called Jagendorf, and what he did basically was to incubate chloroplasts at a high pH, so the inside was a highest pH, and then you dunk them into a low pH and no light at all, and you got ADP and phosphate, and they made ATP because you created a pH gradient, and he showed that just with a pH gradient you could make ATP. No other biochemistry going on at all. So that was a real shock to everybody. So that was a really critical, if you wanted to pinpoint one experiment where you had to say, well, the whole community had to think, oh, wait a minute, maybe he is saying something which has got something to it. You could maybe say that experiment, possibly.
Yeah. So that brings me to this question then, how would someone go about testing something like chemiosmotic theory? What evidence was Dr. Mitchell looking to...
Well, what they specifically did was, and this is what Jennifer Moyle did, so Peter Mitchell, with his technicians there. So he chose technicians who could build bits of equipment. They built a very sensitive oxygen and pH electrode, and what they were trying to do was showing that when you got respiration, you saw protons appear outside of mitochondria, because at the point it was considered irrelevant to the thing. So they spent a long time showing that when you get a respiratory burst, you see protons suddenly appear outside, and then you see them go back in as ATP is being produced. And then if you added, the other critical thing was there were these things called uncouplers, which had been discovered actually in Russia. And actually the Russians were very supportive of Peter Mitchell, because again, he wasn't treading on any toes there. So these things called uncouplers had been discovered a long time before the chemiosmotic theory. And what uncouplers do is they uncouple ATP synthesis from respiration. So you can get respiration going really rapidly, but you don't get ATP synthesis. And of course the biochemists were looking for some way in which these chemicals stuck to the protein that did all this and somehow sort of flipped something off it. So Peter Mitchell investigated whether when you saw these protons come out, if you added an uncoupler, you saw them whiz back in again. So it showed that the uncoupling was due to removing this proton motive force rather than some classical biochemical type thing. So just to warn you, in the newspapers, about every 3 months there's a report of somebody eating a chemical called dinitrophenol, DNP, that you can buy on the black market, okay? It's a slimming agent, okay? And it's true, it's a very good slimming agent. It doesn't matter how much you eat, you respire and you won't make any ATP. So everything is just, you heat up instead, right? So everything goes to heat rather than to metabolic processes. So this was first discovered as being very dodgy in the First World War, because DMP is also a component of ammunition. And they found that the people were getting very ill in the ammunition factory because DMP can be absorbed through your skin. And eventually they found, these people were heating up, and you get multiple organ failure eventually. But unfortunately, these pills are still being sold by disreputable companies, and very occasionally you see someone has died from the slimming agent DNP. I saw it just a month ago again, some teenager had been taking these things. So yeah, don't take, if ever you need to slim for some reason, don't take an uncoupler.
In your lecture about Peter Mitchell, you mentioned that he was right about a lot, but not everything.
Yeah.
Can you elaborate on that?
Well, he was wrong about Morton Wickstrom's thing, for a start. He was also wrong about the ultimate mechanism of ATP synthesis. So getting back to the fact that he really had this very abstract idea about how chemiosmotic systems worked, which is that he thought that the proton motive force impinged directly upon the chemical reaction that was occurring. Okay, by that I mean, for example, if you imagine ATP synthesis, okay, the way that Peter Mitchell thought about it is that what happens is the ADP and the phosphate come together in the membrane, close to each other, and in order to make ATP, to make a phosphate add to another phosphate, you've got to remove water from it. It's a dehydration reaction. You remove an OH from one phosphate and a proton from another phosphate, and then you get the POP bond. That's how it works in chemistry, dehydration. So what Peter Mitchell thought how it worked, which was a great idea, was that here you've positioned the phosphate and the ATP in the membrane, so one side of that pair just sees a very acidic pH. So in other words, it really wants to drag a hydroxide away. And the other side sees a very alkaline pH, so it really wants to drag, so it's dragging a proton this way and a hydroxide that way. It seems like a really neat idea, right? You think, yeah, it's bound to work that way. And right up until the end, Peter Mitchell's mechanisms for ATP synthesis involved a direct contact between the proton motive force and the chemical species which were changing. And that's one example for ATP synthesis, right? You think, well, yeah, it's bound to work that way. It doesn't work like that at all. So John Walker in Cambridge, who sort of who figured out, partially figured out anyway, the ATP synthase. I think I mentioned it in my lecture. It's a very complicated molecular machine. So that what the proton motive force is doing is making this thing rotate in the membrane, and then there's a big protein structure which then rotates in the subunits, and the subunits are being forced to undergo conformational changes at a distance. So ATP is actually being synthesized hundreds of angstroms away from where the proton motive force is actually acting. Peter would have hated that really. But unfortunately, the structure came out after he had died, which was a real shame because I would have really liked to have seen his comments on it. He always wanted the ATP synthase to have somehow be far closer to the membrane than it looked to be, so that he could have his direct mechanism. And this is why he didn't like Morton Wickstrom's mechanism, because he couldn't think of such a direct mechanism. Well, until he did think of such a direct mechanism, which was a very clever one, which I won't tell you what it was, which was getting back to what I said before. That's when I'd arrived there, and the first thing I did, well, I say the first thing, I really sort of 2 years of reading the literature, so I was sure what I was doing. We showed definitively that it wasn't working that way, because on a scale of 1 to 10, well, then you take complex 1, which is this other enzyme in mitochondria. That's very non-Mitchellian as well, because there you've got this huge L-shaped structure, so the chemistry is occurring here, and the proton transfers are occurring down here by a mechanism which is still completely unclear, actually, at the moment. So it's definitely non-classical Mitchellian there. So yeah, he was wrong about quite a few things. I mean, his other favourite thing was economics, besides all these other things, and so and fairness of economics. I don't know if I mentioned, he started paying people at one time in these glins, which were silver units that he'd minted, that people could take to the bank in change for money. And of course these kept up then with inflation, because the price of precious metals was going up at the same rate as inflation, whereas if someone was paid a salary and their salary stays constant, of course they're getting less money each month because inflation is going up. So he decided to have an inflation-linked system, which was this slightly bizarre thing of making silver coins instead. But what he really got wrong there was what was happening to the precious metals market, because at one point while he was doing this, the bottom fell out of the precious metals market and everyone's coins were worth far less than they were the month before. So he got that wrong. But it was a good idea in abstract, again. I mean, that's one of the themes of the things, you know. His ideas in abstract were very solid. And that's true for science generally. It doesn't matter if you come up with something and it's complete and it turns out to be wrong, as long as it was a sensible possibility in terms of the basics, because it has to be tested. And of course you can't know whether it's right or wrong if it's a new idea until you do test it. So there's nothing wrong with being wrong in science, as most science does end up being wrong in the long run. But that's the way the science works. So never worry about proposing something wrong. Just make sure that it's a sensible thing to propose in relation to the basics. Because you don't want someone to say, oh, but wait a minute, this has already been disproven by this very basic thing. As long as it's sensible within the framework of basic science, someone's got to test it. And if you're lucky, it's right.
So are there any major challenges that still have yet to be conquered regarding chemiosmosis?
Well, people are still trying to make biomimetic systems nowadays, to replace things. So one thing which hasn't been done is to make a biomimetic chemiosmotic system properly. Some friends of mine are trying to do that. That's quite a hard thing, to design something in which can do the same things as the chemiosmotic machines. But I think we still don't fully understand the extent of chemiosmotic processes in diverse organisms. So bacteria, for example, have such diversity of electron transfer chains. We're still not sure which bits are chemiosmotic and which are not. And it is quite important to know that stuff from energetics point of view and controlling and targeting sites in bacteria and so on. But in terms of the basic chemiosmotic theory, I think it's firmly established now in science. Mechanisms we're still not sure of. The mechanisms are far, far more complicated than Peter Mitchell ever envisaged in the first instance. Some of his stuff was correct for that, but I would say the majority wasn't correct there. But nevertheless, he supplied the abstract formulation within which whatever you decide is the actual mechanism it has to fit, sort of thing. So I think there's still a lot to learn about how widespread some of these chemiosmotic things are, and we still need to understand the fundamentals of some of it. Even with something like, for the enzymes where we have crystal structures, depends how far you're going with understanding the relation, the physics of electron transfer and proton transfer. That's still a very big area as to how these things work on a fundamental physical sort of level. So yeah, there's still a lot to learn, but speculating what's the next big thing, of course, if I knew that, I would be madly working on it to know what it's going to be. You never know until you've gone down a certain path as to where quite something is going to end up.
So you said if you knew that, you'd be madly working towards it, but
Yeah.
What are you actually working towards right now? What is some of the current research going on in your lab?
We're still trying to understand the fundamental mechanism in cytochrome oxidase. It is getting around to looking at the structures, to how the protons move through the structures of the enzyme. So most of my lab is geared up for spectroscopy, and in particular mid-infrared vibrational spectroscopy, where we can actually see protons jumping on and off amino acids. So that it's the only method you can use to actually see the protons. To see in the electrons you can see with UV visible spectroscopy, EPR spectroscopy, lots of things. But to see protons, you can't even see protons in a crystal structure, right? The protons are they haven't got enough electron density. So you never see a proton in a X-ray crystallographic atomic structure. You have to guess where the protons are. But not only that, an X-ray crystal structure is a static structure, whereas we need to know what's happening on a sort of microsecond time scale. So we're trying to do that with spectroscopy. So fire electrons through the system, see where the protons go. We're trying to do it, and this is not me myself, my collaborators. I'm only expert in a few things. If I need to do something else, I find someone else who's expert and then collaborate with them. So actually I've got one picture I can show you. It's now possible with supercomputers to take even a very complicated structure like cytochrome oxidase, 13 subunits, thousands of atoms, and you can stick it in a membrane. Do I have it up here? So this is something actually which is ongoing, and we were discussing this partly in the meeting I was at last week. Here's a structure. There's part of cytochrome oxidase. It's in a membrane. I've removed the lipids at the moment. We've got the lipids in there as well, and then we've got two water phases with ions in it. So this is simulating. This is a computer model of cytochrome oxidase stuck in a membrane with two aqueous phases either side. Okay. And there's about several 100,000 atoms in that in total. Okay, so then we do quantum mechanical calculations to work out what the electronic distribution is around the metal centres. We use infrared spectroscopy to tell us what the pKs are of the amino acids that matter. And then we use the supercomputer to tell us how this all moves on a nanosecond to microsecond timescale, so that we put an electron in and then we ask the question, what is happening to the water molecules and the protons within the structure? How are they distorting as the electron moves through the structure? So we get this to predict for us what we need to look for in things like our infrared experiments, because in infrared we're able to see individual water molecules actually moving in the structure. We're trying to understand where they are and how that relates to the what's called PCET, proton coupled electron transfer, which is a physics topic, to understand the forces involved in that. So what we're doing in my lab is trying to combine this sort of supercomputing type of stuff to predict things that we can actually then go and measure in the lab in order to try to understand these mechanisms on a more fundamental basis. And then, because you need to get funding for stuff, okay, and getting funding for basic stuff like this is much harder than getting funding for stuff which is medically related. But you're medical science. So the other half of stuff in my lab, we use our very advanced technology for medical applications. I can show you some of that if you like. So with infrared vibrational spectroscopy, it's a very advanced technique. We've modified it so that we can look for single amino acid changes in proteins and so on. But we can also use it for biomedical applications. So I'll show you a machine in there. You just put your finger on and we can see some of the stuff in your finger. So you can tell for certain disease states what things are. And so I've just had 2 students, they've just finished now, but we've been intercepting biopsy samples from the cancer clinic, and we do a 30-second vibrational spectrum, and then we send the biopsy sample back to the histologist to do their standard analysis of to grade the cancer level, and then we compare our results, which take 30 seconds, with their results that take 2 weeks, and we get a good correlation actually. But the problem is we get something where we disagree with them, and the question is who's right, and we're not sure about that, because actually histological examination of cancer biopsy samples is a very difficult thing. It's very user dependent. So normally they have 2 histologists independently grade a cancer sample, and if they disagree with each other they get a 3rd one in, but the amount of disagreement is quite large, so we're still trying to deal with that. But my last 2 students, they're just in the process now of forming a company in order to develop this further. And I'll show you a machine that we've been running in the kidney unit up at the Royal Free Hospital, run by nurses, where they're putting patient urine samples onto the thing, where we're looking for diagnostic materials within the urine that can't be seen by what normal clinical practice. So we're just about to publish a paper now on cystinuria, which is a relatively rare kidney disease, where it's caused by cystine. It's a genetic abnormality actually. You get cystine in the urine, which is rather insoluble, and it forms microcrystals that can form nucleation sites for kidney stones and so on. So it's a quite nasty disease, but it's very hard to pick up clinically. So we can see that in the patient urine sample, so that you can do again a 30-second sort of test and see what the cystine level is. So that sort of thing. So we're doing practical stuff. But the fun stuff for me is not that. The fun stuff is this.
Sorry, what are you exactly measuring with the machines and the finger for cancer?
No.
Or?
We could do. We can only look into the immediate, the first few microns, because the beam only penetrates the first few microns of whatever we're doing. So if you've got a deep cancer, I mean, there's nothing we can do about that at all. But for example, in the future, if for example you had an organelle cancerous region that you wanted to define where the cancerous region was, potentially you can make a probe version of this thing and just touch it onto the organ rather than have to take a biopsy sample. At the moment, with our project there, this is esophageal cancer. They physically have to take biopsy samples down the esophagus, which is a fairly horrendous procedure to begin with. So in the finger, we can just, for example, we can see your glucose level, for example. We can see if you've been drinking too much alcohol. We can see various metabolites.
So it's bloodstream then?
If the blood vessels are close enough to the surface, we'd be able to see stuff in the blood as well, actually.
Okay.
Yeah, so actually if you did it on the ear by here, where the blood vessels are very close, or within the mouth on the top of the buccal cavity, you could see a lot more. In the finger, of course, we've got a very thick layer there, so you're not seeing very much. I just use it just to show people the what the thing can do. But in terms of looking at your finger, there's only a certain number of things you can see. But in other parts, it would be okay. Or for example, if you had a melanoma, that's on the surface, so that is very powerful for detecting melanoma. So, like all these things in science, there's pluses and minuses to it. You have to find the way to go through. So the urine stuff is good, but of course that's non-invasive, really. No one minds giving a urine sample. Or we could use a blood sample. You only need microliters of the stuff. You need a tiny, tiny amount. And the other important thing is it's non-destructive. We're not doing anything chemical or anything to the sample. So when it comes to biopsy samples, which are of course fantastically valuable because they're so hard for the person to give, there's no way we could have done this research if our method was messing the samples up. The only reason we managed to do the project is because we can make our measurement and give it straight back to the histologists, so that there's no extra material being required to do this sort of thing. And then the other thing that we've been doing with that as well, in here we don't have the equipment on site yet. I've been collaborating with a guy in chemical engineering in Imperial. So with the technology now, you can do the thing microscopically. So you can take a thin sample and you can look with an FTIR microscope and pixelate it, and so you can get an infrared spectrum of every point on the spectrum. And then what you can do is you can then, if you understand the infrared spectra, you can make a biochemical map. So you can map glucose, or you can map collagen, or you can map DNA. So you can map different things, which then highlight the regions which are cancerous on a cell-by-cell sort of basis. Because the problem in using a biopsy sample for cancer diagnosis, if you're trying to do a one-off global measurement, and of course that sample may only have a small region of cancerous cells in it, then you're averaging out the cancerous cells with the non-cancerous ones. But the thing is that to introduce FTIR microscopy into a clinic, that's years off, because you need specialists. We're trying to go down the simple route of introducing something that a nurse can actually operate in a hospital.
So you've been doing a lot of diverse kinds of work, and you said earlier how Peter Mitchell gave you a lot of freedom to do what you wanted in his lab. Is that something that you've been trying to carry forward in your own lab?
Yeah. I would be delighted if students went off on their own direction to something completely different that I haven't even thought of. That would be great instead.
Well, thank you so much for your time.
Okay, thanks. Nice talking to you.
Attention.
I'll give you a brief view of the lab if you like. One of you can put your finger on one of our machines and see how you're doing.